In the process of eating protein, when you eat meat, your body sometimes has to get rid of excess. Proteins are made of amino acids. This is by processing excess amino acids and converting them into molecules that are used in respiration. So what I want to go over today is the amino acid alanine. So the amino acid alanine, this is part of the carbon 3 family. Okay, which consists of so you got alanine, which is so the alanine amino acid is identified by the CH3 there, the cysteine by the cysteine by SH, the serine by the OH group there, which is after that CH2, and the threonine by this attachment. Okay, we have hydroxyl group, a carboxyl group, and an ammonia group there. Okay, 3 different groups: OH, COO, and the negative NH3 plus, and the amino comes from the NH. This is the alpha carbon. So it comes from the NH3 at one side and the acid. This is the acid group at the other. So amino acid. That's the name. Amino acid comes from it being amino acid, and you got the groups identifying them: alanine, serine, threonine, cysteine. So this is the carbon 3 because in each case there's 3 carbons: 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay, this is the carbon 3 family of amino acids. So returning to the reaction alanine, I should say the enzyme that actually catalyzed this is called Alanine Transaminase. Alanine Transaminase. So you've got plus alpha ketoglutarate, which is also called alpha ketogluteric acid. Okay, so you got the carboxylic acids group at both ends actually, but you got that's the keto there refers to this, so that's the alpha carbon, that's the carboxylic group, and that's another one, so alpha keto and then glutarate. But that'd be the alpha carbon. So this is an equilibrium, and then you got alanine transaminase, and then it becomes, so that's alanine, and this is alpha ketoglutarate becoming that's pyruvic acid plus okay, there are hydrogens on these, but I'm not drawing them in, okay, so you normally don't. This is L-glutamic acid, the glutamate. The L refers to it being an isomer, okay? And the way light is, the polarization of light is altered by the isomer when you shine light through it. It's a way of identifying it. L and D. You get the L and D forms. Okay. So that's the reaction, alanine plus alpha ketoglutarate. The amino alanine transaminase, the amino group here is transferred to the alpha ketoglutarate and becomes that goes there, okay? It attaches to there and goes there, and the alanine becomes pyruvic acid, okay? So the production of pyruvic acid, that can lead to glycolysis. That can go as a glycolysis, okay? So by converting excess amino acids into pyruvic acid and L-glutamate, this can be shuffled off to glycolysis for the creation of ATP, which we know is main glycolysis, okay? Energy. So you've got, so these carbon skeletons, you get there's 20 amino acids, which are sort of generally in diet, in the body in diet. Of the 20 main amino acids, okay. The carbon skeletons are funneled into seven molecules, okay? So the 20 actually made into only this number. 7, which consist of so these ones are acetoacetyl. Not quite sure where that comes in, I can't quite remember, but these ones are part of the Krebs cycle. Okay? Can't remember where that comes in, but these ones are part of the Krebs cycle. So that one may be, but I can't remember where that one comes in at the moment. So yeah. So the alanine transaminase catalyzes the transfer of the NH3 from the L-alanine to the alpha ketoglutarate, okay? And because pyruvic acid is made, okay, these reactions are called glucogenic reactions, okay? They're glucogenic reactions, okay? And also, yeah, and through these reactions, alpha ketoglutarate, succinol-CoA, fumarate, and oxaloacetate are also made, okay? So these are made, these molecules are made in the glucogenic reactions. So the net synthesis of glucose from alanine happens because the Krebs cycle intermediates and pyruvate can be made into phosphoenopyruvate. So yeah, so this kind of can be summarized. So yeah, so I should say overall, you get alanine, glycine, serine, threonine, and tryptophan. Okay, these lead to pyruvic acid. Okay, which then goes into the Krebs cycle, oxaloacetate, and then citrate, and then alpha ketoglutarate, and then succinyl-CoA, and then fumarate. Okay, and then back around here, but from oxaloacetate, that can lead to phosphoenopyruvate. Okay, also known as phosphoenopyruvate, which can lead to glucose. Okay, so the net synthesis from alanine happens because these Krebs cycle intermediates can be made into phosphoenopyruvate. But in mammals, this is lacking. Okay? So we've also got so yeah, this shows the fates of the carbon skeletons of the carbon 3 amino acids in the glucogenic pathways. Okay, so yeah, oxaloacetate can lead on to phosphoenopyruvate and glucose. Also, I should mention the amino acids. The amino acid, serine, can be converted to pyruvic acid plus NH4 plus by serine dehydratase. Okay, and cysteine can be made into pyruvate by several paths. It can be made into pyruvate, and its S atom, the sulfur. Remember, the serine looks like the cysteine looks like this. So the sulfur atom can emerge as in either H2S or SO3 2 minus or FCN. Okay.